Welcome to Let's Get to the Marks. We're doing question 1, AQA Biology, Paper 1, 2023. So give the three structural features found in all virus particles and describe the function of these features. So we basically need to list the names of three features. So the first one I'm going to do here, we're going to do the attachment proteins. So just drawing attachment proteins. Of course, this is what the virus uses to attach to a host cell that it's going to invade. So we've got the attachment proteins, and that will get you a mark. Now, the second mark, you're going to need to name the other two things. So, what other two features do we have in a virus? Well, inside of the attachment proteins, we have this structure called the capsid. So, hopefully, you know the capsid. I've drawn it here in orange on my little make believe virus, and that's the capsid. Now, within the capsid, you'll find these little wiggly purple things I'm drawing. What's this? This is genetic material. You call it the genome or the DNA or RNA because you have different viruses. Like COVID was an RNA virus. So you get different styles of virus. Some use DNA, some use RNA as their genetic material. Right, now we have to explain the function of just one of those features. So I'm going to do them color coordinated. So first we'll explain the function of the attachment proteins, which I've already mentioned. They allow the virus to attach to receptor sites on the host cells. A specific example you have to know is that the HIV virus attaches to the CD4 receptors on T cells. Anyway, let's look at the of the capsid? Well, it's there to protect the genetic material, or you could say protect DNA, RNA, or the genome. Now, what's the genetic material for? I'm writing that in pink, purple, whatever. Well, it codes for proteins, specifically viral proteins. And that's DNA and RNA always code for proteins. But in a virus, it's going to be viral proteins. Right, let's look at 1.2. Explain why viruses are described as acellular or non-living. Well, what does acellular mean? It means not a cell. So viruses are not cells. What's the evidence for that? They contain none of the structures or organelles that you find in cells. And you could name a structure. A virus won't contain a mitochondria. It doesn't have cytoplasm. It doesn't have a cell wall. It doesn't have cell membrane. Something that all cells contain would be a cell membrane or cytoplasm. So that would be a good thing to put. They do not have those. Not all cells have mitochondria, so you probably shouldn't put that. Okay, now what other evidence? Well, we've got why a virus is described as non living, not alive. Well, they don't carry out metabolic processes like digestion, they can't respire, they can't reproduce, they can't replicate. So we've got lots of reasons why we call them non living. They can't do the processes that living things do. So you need one thing saying why they're not cells, and then one of these to say why they're not living. Okay, let's move on to 1.3. Give one reason why antibiotics are not effective against viruses. Well, there's plenty of reasons why they're not effective. Antibiotics normally target enzymes, the ones that build the cell wall of a bacteria. So viruses don't really contain enzymes, but viruses don't even have a cell wall. So they don't have murine, peptidoglycan, or glycoproteins. Either of those are the chemicals found in the cell wall of a bacteria. So there's none of this stuff that the antibiotic can disrupt. Some antibiotics disrupt metabolic pathways inside the bacteria, and of course, viruses don't have any metabolic processes. All right, guys, hit the like. That's the end of the first question. Let's go.
If you push the button,
Would you like it?
Chitin is a polysaccharide. The chitin monomer is a beta glucose molecule with one hydroxide group replaced by NHCOCH3 group. NHCOCH3 group can be represented by NAC. Figure 1 shows the monomer. This forms chitin and the chitin polymer. I've labeled those here. Chitin is a similar structure to cellulose. Use figure 1 to describe 3 ways the structure of chitin is similar to the structure of cellulose. You can see the structure of cellulose up here. Okay, both cellulose and the chitin in the figure 1 have 1 to 4 glycosidic links between the beta glucose molecules. So that's going to get us 1 mark. They both got 1 to 4 glycosidic links. Don't say 1 to 6. That's the branching. Now, both these molecules, both the cellulose in the picture and the chitin, have no branches coming off of the 6th carbon, so no 1 to 6 glycosidic links or no branches. They're both linear molecules. And every other molecule is reversed 180 degrees. And an additional point, both are made of beta glucose. It tells us in the stem of the question that chitin is made from beta glucose. You have to know off by heart that cellulose is also made of beta glucose. And here we go. Let's move on.
Keep moving on, keep moving on, keep moving on, keep moving on.
Okay, 2.2. Chitin keeps the trachea open in the tracheal system of gas exchange in an insect. Gas exchange does not occur in the trachea. Explain the importance of one adaptation of the gas exchange surface in the tracheal system of an insect. So, first of all, we need to draw out the tracheal system, the breathing system of an insect. Okay, here's where my D grade in GCSE art comes into handy. So this is the muscle right here. The muscles of the insect. So you'll find them in the legs, the abdominals, and various regions on the insect. Now, what I'm drawing here is the tracheoles, those tiny little branches leading into the muscle, and then the trachea, which is this long tube here. And the holes at the end of the trachea are the spiracles that actually allow ventilation of gases into and out. So the trachea have these rings of chitin, shown by these black lines, and the tracheoles are a highly branched network, a little bit like the bronchioles that lead to the alveoli in our lungs. So if you think of an insect, these blue circles here are the spiracles, the air is ventilated in, and then it moves down the trachea, which has got rings of chitin, and into the tracheoles, and finally into the muscle. So what is one of these adaptations and the importance? So we could say, well, the tracheoles are only 1 cell thick. What does that do? Well, just like alveoli or the villi, it creates a short diffusion pathway. So that's your explanation. Your description or adaptation is that they're 1 cell thick. So I'm going to put an A here. And the explanation, short diffusion pathway. Let's look at another aspect of this breathing system or the tracheal system of the insect. So another another adaptation of the tracheoles is that they enter directly into the muscle. Again, this creates a short diffusion pathway. So you can see that here, the end of the tracheoles leads directly into the muscle tissue, meaning there's a short distance for the gases to diffuse into the actual muscle cells. Okay, that's the adaptation and explanation. Final one, the tracheoles or tracheoles are highly branched. What does that do? That creates a large surface area as they're a massive network, a bit like the alveoles and the bronchioles and the lungs. This allows a large surface area for rapid diffusion. So just like humans have millions of alveoli, insects have millions of tracheoles in a large network, creating a large surface area for rapid diffusion. You only need one of these lines here, just an A with an E, an adaptation and explanation. Let's look at 2.3. Lignin is a polymer found in the walls of the xylem vessel in plants. Lignin keeps the xylem vessels open as one continuous tube. Explain the importance of the xylem being open as a continuous tube. So we're not really being asked about why lignin's important. We're really just being asked, why is it important that the xylem is a continuous tube? Why is that important? We've got the roots here at the bottom in brown, and I've got the xylem tube going up the stem, and then we've got the leaf. Of course, you have evaporation by transpiration in the leaf. What does that do? That creates tension. Okay, so the transpiration of H2O molecules through the stomata creates a tension, a pull or a force or a negative pressure pulling water up the xylem. We call this tension. Okay, so that's going to get you a mark for explaining where we get tension from. Of course, the next thing to consider is that in order for water to move up the xylem, they're going to have to be able to stick together. And water molecules are polar. And what that means is not that they like the Arctic, but actually they're attracted to each other. Okay. Why are they attracted? They have two lone pairs on the oxygen, making it delta negative or partially negative, and hydrogens are partially positive. And what we get is hydrogen bonds, forces of electrostatic attraction between the partially negative oxygen and the partially positive hydrogen. And so they move up in a continuous column because they're attracted to each other. If you severed that column, if you severed those bonds, all the water would fall down. So they form a continuous column of water, unbroken column of water, moving up the xylem. And so that's important that the xylem has no end walls and it's free for the water to move. And this is by the theory of cohesion tension. So you're going to get all your marks for these points here. And that's really the end of that question. Now, I mean, if you break the water column, it's going to really interfere. So, like making a hole into the stem of a plant, which could potentially break the water column, is going to be very detrimental to a plant. Okay, there's your 3 marks. Remember to hit the like, subscribe, check out my other videos.
Make sure you pause the video and have a go at the question, then follow the walkthrough to mark your work. So 3.1, the human disease malaria is caused by infection with a single-celled eukaryotic organism. Figure 2 shows a diagram of the Plasmodium vivax, one of the species that can cause malaria. Structures containing enzymes that allow P. vivax to enter human cells. Other than the Golgi apparatus, name one structure in Figure 2 which shows the P. vivax is a eukaryote. You can see the Golgi apparatus here. So some of the structures you would only find in eukaryote, like mitochondria here. So mitochondria, it's got a double membrane or an envelope with cristae on it. Then we've got the nucleus, which has a nuclear envelope, so another double membrane bound organelle. Any double membrane bound organelle you can see in this diagram. We've also got the rough endoplasmic reticulum, which is not double membrane bound, but not found in prokaryotes, so only found in eukaryotes. Now, some people might make the mistake of saying smooth endoplasmic reticulum. That will not get you a mark because that is not shown in Figure 2. And the question specifically asks something that is shown in Figure 2. You would only find smooth endoplasmic reticulum in a eukaryote, but it's not shown here. So that's not one of the possible answers. Now, you do find ribosomes that I've circled here. You do find those found in both prokaryote and eukaryote, but specifically these are ATS ribosomes. So that will get you the mark as well. But just no smooth endoplasmic reticulum. Now, some people get the Golgi apparatus mixed up with the smooth endoplasmic reticulum. They have a slightly different shape. The Golgi apparatus is more C-shaped, and you will have little vesicles arriving and budding off of the Golgi apparatus. So 3.2, describe 2 functions of the Golgi apparatus in a eukaryotic cell. So we can see here they produce vesicles for transport. Of course, things go to and from the Golgi apparatus. I'm circling here. They go to the membrane for exocytosis, etc. It's also the site of packaging. Oops, I put packaging and modification of proteins. So packaging or modifying proteins. This can be adding a glucose molecule to make a glycolipid, or a lipid to make a lipoprotein. There's all sorts of things we can do, but they also used for modifying lipids. We could make a glycolipid. So by adding a glucose to a lipid. Okay, so we can stick things and modify fats, lipids, or proteins here. It's for packaging and modifying those, also for producing vesicles for transport. All right, let's move on to 3.3, which is the next part of this question. So here's the stem. P. vivax evolved from a common ancestor in Africa. As humans migrated around the world, new strains of P. vivax evolved. P. vivax is now extremely rare in Africa, but there are several different strains of P. vivax in other parts of the world. Figure 3 shows a phylogenetic diagram of the evolution of these different strains. So you can see there's common ancestors at every point on here. And the question 3.3 says, what does Figure 3 suggest about the order of human migration out of Africa? Well, if we look at the African common ancestor there, which is now extinct, go to the nearest point and write 1. that is the closest ancestor to the common African ancestor. Where's the next nearest point? It's over here. 2, and that's the European strain. Then 3, the Indian strain. Then 4, the Central American. And then 5, finally, is the South American strain. So these phylogenetic trees or diagrams can show you how closely related things are. So follow the numbers I've written and it will go India, Europe, East Asia, Central America, and South America. And there we go. That's your mark. Let's move on to 3.4. Now, 3.4 is a calculation. So there are an estimated 229 million cases of human malaria worldwide per year. 94% of these cases are found in Africa, but are not caused by P. vivax. P. vivax does cause 61% of the cases of human malaria outside Africa. Use this information to calculate the number of cases worldwide caused by P. vivax every year. You can see I'm underlining key points. You can use a highlighter if you like. It's just good exam practice to get in the habit of highlighting, circling key information. That way, when you're in an exam, it may stop you from missing something really important. Even if when you first start doing it, you highlight loads of useless information, at least it helps. Right, 94% in Africa is not caused by P. vivax. So that means 94% of the 229 million cases worldwide are not caused by V. vivax because they're the African ones. So we're going to go 100 take away 94 is 6%. Only 6% of the total can possibly be caused by P. vivax. So we go 0.06 times 229, or if you like, 229 divided by 100 times 6. And this equals 13.74 million. Now, it told us those are the amount of cases that happen outside Africa that could be caused by P. vivax, but we were told that only 61% of these were caused by P. vivax. So now do 0.61 times 13.74, and you're going to get something like 8.38 million. Round it down to one decimal place to 8.4 million, and that's your number of cases of malaria. Orsomenus. We're nearly through. We've got a 4-mark question now on natural selection. So, 3.5. In Africa today, most of the human population are resistant to malaria caused by the P. vivax. Use your knowledge of natural selection to explain why this resistance is so common in Africa. So, back in GCSE, this 4-marker would have been a mutation causes a change in a gene, makes you more likely to survive, reproduce, pass the gene on to the offspring. There's a slight difference at A-level. We must use the word allele. So mutation causes a change in a gene which produces an allele which makes people more resistant to malaria. We do the whole more likely to survive because they won't catch malaria, pass on this allele to their offspring, reproduce and pass on to their offspring, and then we need to finish with the A-level will increase the frequency of the allele in the population. So that's the kind of difference between the GCSE. We're using the word allele a lot, and we need to end by saying the frequency of the allele is increased in the population. So you do the whole more likely to survive, more likely to mate successfully or reproduce or breed, produce offspring that contain the allele. At GCSE, you'd say this is repeated over many generations, which we can still say here, increasing the frequency of the allele in the population. And that's the end of question 3. So I hope you did well. Bish bash bosh, I think that was an 8 or 9 marker. So there we go, all done. Subscribe, share and like, helps me with the channel. Let's get to the marks. Okay, going through question 4. Some hospital patients suffer from diarrhea caused by infection with the bacterium Clostridium difficile. The C. difficile bacteria releases toxins. These toxins cause diarrhea. The toxins damage the cells lining to the ileum, causing them to lose their microvilli. The damage to the cells reduces the absorption of the products of digestion and reduces the absorption of water, resulting in diarrhea. Explain why the damage to the cells lining the ileum reduces the absorption of the products of digestion and why this reduces the absorption of water. So let's answer this question. You might want to pause the video and have a go. Have a go on some scrap paper. See if you can write down 3 things that will get you 3 marks. Okay, so let's imagine this is the villi And these are the cells behind the villi. And then these tiny little black lines that I'm crustily drawing, they're the microvilli. Okay, and of course, you've got to ask yourself, it tells you in the question that microvilli are damaged. So what's that going to affect? What do the microvilli, how does that increase the function of the epithelial cells in the ileum of our small intestine? And of course, what they're saying is that there's toxins released from this bacteria that actually damage all those microvilli. So I've wiped the microvilli out and the toxins have done that. How's that going to affect the functioning of the small intestine? And of course, the toxin damages the microvilli and reduces the surface area. And there's your first mark. Okay, so we've got the surface area is damaged, well, is reduced by damaged. We also have potentially protein channels or protein carriers damaged, things like aquaporins, things like your glucose, sodium co-transport proteins. All of these things could be damaged. All of this results in the less uptake of solutes of the products of digestion. So less glucose, less amino acids, less fatty acids and things being taken into the cells lining the lumen of the ileum. What does that do? Well, that raises the water potential of the cells surrounding the ileum because they're not getting glucose and amino acids in, which would normally lower the water potential. So this raises the water potential of those cells. So what happens then is water moves down a water potential gradient out of the cells around the ileum into the lumen of the ileum, causing diarrhea. So look at this little model here. Normally glucose and the products of digestion cross the villi and enter the cells surrounding the ileum. What does that do? That normally lowers the water potential around those cells. However, in this situation, and so normally then H2O would follow from the lumen by osmosis into the cells around the ileum and then on into the blood. However, we have a bit of a reverse situation going on. The products of digestion this time cannot cross because these protein channels have been damaged, the surface area has been reduced, so they just can't diffuse or move across quickly enough into the cells around the ileum. And what happens then is the water potential is raised in the cells around the ileum, and therefore the water moves in the opposite direction. It moves down a water potential gradient from a high water potential in the cells around the ileum into the lumen of the ileum, which causes diarrhea. And so you get a nice easy 3 marks here for explaining that the water potential in the cells around the ileum are raised and the water potential of the lumen is lowered. Osmosis, water moves into the lumen, causing diarrhea. There we go. Yuck, yuck, yuck. Right, let's look at the next question. Not all patients in hospital with C. difficile develop diarrhea. Scientists measured the antitoxin antibody concentration in hospital patients with and without difficile infection. They measured the antitoxin antibody concentration. So here we have patients with C. difficile infection but no diarrhea. You can see they have a high percentage of antitoxin antibody concentration. Whereas patients with C. difficile and without infection, without C. difficile have a kind of concentration of 1, and then the people with the lowest concentration of the antitoxin antibody, they're the people with diarrhea. The scientists suggested that the antitoxin antibody could be given to some patients as a form of passive immunity. Which patients should get it, and how would this passive immunity work? Well, if they're given the antitoxin antibody, it's going to neutralize the toxin produced by the C. difficile. So that explains how this passive immunity would work. They would then have the antitoxin in their body, which would neutralize the C. difficile's toxin that's damaging the lining of the ileum. So no longer would they get a damaged lining of an ileum. It also may encourage phagocytosis of the C. difficile, as the antibodies would join to the C. difficile and do agglutination, and that would encourage phagocytosis by making a big clump or clumping of the C. difficile. Now, which patients should you offer this treatment to? Well, there's no point in giving it to the people who don't have diarrhea because they have a high concentration of antitoxin in their blood. The patients that are infected but with no diarrhea, they already have a high antitoxin antibody concentration. So we want to give it to people who have diarrhea because they're the ones who have a low concentration of antibody antitoxin in their blood. Okay, 4.3. To be used as passive immunity treatment, the antitoxin antibody would be injected if it was given by mouth. So just why it would be digested. So we need to say, why would it be digested? Well, antibodies and antitoxins are obviously proteins. So they're made of amino acids joined together by peptide bonds. So how do you digest a peptide? You've got to use your A-level content. You can't just say protease digest protein into amino acids in the stomach, pH 2, all of that business. That is GCSE. So what we need to consider here is that you have exopeptidases, and they break off the end amino acids by hydrolyzing peptide bonds on the outside of the polypeptide. And then we have endopeptidases that hydrolyze peptide bonds inside the polypeptide, so within the chain. And so there you go. You just really need to describe the action of those two different types of enzyme, which is your A-level digestion of proteins content. So endopeptidases hydrolyze the bonds within the polypeptide. Exopeptidases hydrolyze the bonds at the end of the polypeptide chain that are on the outside of the polypeptide. And you're using the word hydrolyze, you're going to get a mark. You're talking about different peptidases and you're mentioning the peptide bond is being broken. So peptide bond and hydrolyze would get you the marks, and then a description of what exopeptidases and endopeptidases do will get a mark. Lovely jubbly. A student investigated the use of cinnamon oil as an antimicrobial substance. She investigated the effect of cinnamon oil on the growth of 5 different bacterial cultures grown on agar plates. The student added 100 millimeter cubed of each bacterial culture from its glass bottle onto a separate agar plate. She spread each bacterial culture evenly over the agar using a spreader. Describe the aseptic techniques she should use for 3 marks. Okay, you should try to list off 3 aseptic techniques that you can think of. If you can do 4 or 5, even better. That way, if one's wrong, they may allow it. All right, guys, pause the video, have a go, then unpause, watch the walkthrough. Let's go. Okay, I'm going to go through the answers now. So one aseptic technique we can use is to work close to a heat source, such as a Bunsen burner flame. This will make the air rise by convection currents and take any microorganisms in the air that may contaminate our plate, take them up and away from the area we're working. Hence, protect the plate from being contaminated by microorganisms. Another thing is the person carrying out the experiment, the student in this case, she should clean her hands. You can just wash them with soap or antiseptic solution or antibacterial hand wash, something that will kill microorganisms or get rid of them on the hands so they don't contaminate the agar plate. Another thing to do is sterilize the spreader before use, or the pipette, or any of the equipment, so that microorganisms on the equipment are killed and cannot contaminate our agar plate. Flame, pass the neck of the bottle through a flame. So you can say flame the neck of the glass bottle. That will kill any microorganisms on the glass bottle around the neck. And another technique is to only lift the lid on your agar plate at an angle. Again, this prevents contamination or reduces the likelihood of contamination by microorganisms in the air. Okay, let's move on to the next question. Each agar plate, the student cut a well or a hole into the agar. The well had a diameter of 6 millimeter. The student added 50 millimeter cubed of cinnamon oil into the well. Calculate the minimum depth of the well to allow the addition of 50 millimeter cubed of cinnamon oil. Use the following equation in your calculation. Volume of the cylinder equals pi r squared times L, where L is the length of the cylinder. Use 3.14 for the value of pi. Show your working. Notice I've highlighted the key data. Always use a highlighter pen in your exams. It's really important so you don't miss a key command. Okay, they've cut a well into some agar gel. Also, I like to do little drawings as well so I can visualize what I'm actually doing. They've put 50 millimeter cubed of cinnamon oil into this well, and we know the diameter here is 6 millimeter across the well. So we have an equation. Volume of a cylinder is equal to pi r squared times the length. We need to find, we've got the volume because they've told us it's 50 millimeter cubed. We've got the diameter, so we know pi, so we know r. The thing we're missing is the length. The length is actually equal to the depth. So here's our equation, and we want the depth. Now, if you think about a cylinder, the length of the cylinder is equivalent to the depth because, of course, our cylinder is being cut into the jelly, and that means its length across or going down once it's cut in is the depth. So that's what we're looking for, guys. We're looking for L here. So we want to rearrange this. So I want to take the pi r squared and stick it underneath the volume. And I do that by dividing both sides by pi r squared. And that's going to leave me with volume over pi r squared is equal to the length. So now our volume is 50 millimeter cubed. We want to divide that by pi, which we're told here is 3.14. So we can just use 3.14. The radius is going to be 3 millimeter because it's 6 divided by 2, the diameter 6. And we need to do that to the power of 2 because it's squared. And we'll get the answer 1.769. I'd probably round this to 1.77, but there'll be a whole range of answers they will accept. And there you go. That is how to do this calculation. Just a quick video for today. So I hope you paused it and had a go. And there we go. Nice one. Let's get to the marks. Okay, next question. The student kept the plates at 25 degrees for 24 hours. Figure 5 shows what one of her agar plates looked like after 24 hours. So we've got the bacterial culture around the outside, growing on the agar. We've got the well containing the cinnamon oil in the middle, and then we've got this clear zone or zone of inhibition around that well of cinnamon oil. And that's where the bacteria have either been inhibited from reproduction or are dead. So the student measured the diameter of the clear zone with no bacterial growth around each well. She made measurements to the nearest whole millimeter. Table 1 shows the results. We can see we're missing a few results here, so we're obviously going to be asked to fill them in, and that's the median for all cultures and the mean for all cultures. And we've got, with cinnamon oil, we've got a positive control and something called a negative control. We can see with the negative control, none of the bacteria died. Suggest exactly what should be used for the positive and negative control. So with the positive control, there is a clear area around the center. Now, there wouldn't have been any cinnamon oil, but this positive control has killed some of the bacteria because there's a clear area, whereas the negative control hasn't killed any bacteria. So really, with the positive control, we're just proving that the bacteria can be killed, and that creates the clear area. And so for the positive control, we want to use something that is universally known to kill bacteria, some sort of antimicrobial, something that kills bacteria. And then we can therefore show that these bacteria can be killed. And then for the negative control, we can use sterile water. So water that doesn't have microorganisms in it. It's been sterilized with chlorine or UV light. And we can show here that water, the bacteria don't just die under normal circumstances. So there's our negative control. Now, 5.4 says complete table 1. So what we need to do is come down here and work out the median for all cultures. And so we've got to do the median for the cinnamon oil and the median for the positive control as well. So what we need to do is put the results in a list order of largest to smallest. So for the cinnamon oil, it goes like 20, 18, 16, 15, 14, and 16 is the middle number in the list, is number 3, the one right in the middle of the list that splits the top values from the bottom values. Whereas with the second set of results, we have 17, 14, 12, 12, 12. So what splits the top values from the bottom values? It's 12. Right bang in the middle there, the middle result is 12. Now we've got to work out the mean for all the cultures. So you're just going to add 15, 20, 18, 16, and 14 together, divide by 5, you get 16.6. And if we do the same for the positive control, we get 13.4. At least I did anyway. You could round to a whole number, as that's what they've done in the tables if you like, or you could just leave with the decimal like I'm going to. Right, 5.5, the mean plus or minus 2 times standard deviations includes over 95% of the data. So this is important. The mean plus 2 times standard deviations. You could easily miss that and think it just means plus or minus 2. So they're saying the mean plus or minus 2 of the standard deviations. Use the information to consider whether the standard deviations suggest there are differences in the mean are likely to be due to chance. Explain your answer, including at least one calculation. Let's look at the two means. So 16.6 We need to zing up and see what the standard deviation for 16.6 is. It's 2.4. And for 13.4, the positive control, it was 2.2. Yeah, maybe it was 2.2. So it was 2.4 for 16.6, and I believe it was 2.2 for the 13.4. Now remember, we got to double these because it says 2 times standard deviation in the question. So that means we have 2 times 2.4 means we have the standard deviation has been doubled to 4.8. So I'm going to plus 4.8, because what you do with the standard deviation is you add it and minus it and you find the range around the mean. So this means that it went up to 21. What's 4? That's 21.6, 21.4. So it goes up to 21.4, and that is plus 4.8. Now we want to minus 4.8 because it told us that minus 2 standard deviations from the mean, which were 2.4. So we want to take 4.8 off, and that gives me 11.8. Now I'm going to do the same here, but the standard deviation total is different because it was 2.2 times 2 is 4.4. So I'm going to add that to 13.4 and get 17.8. And then I want to minus 4.4. And this is the range around the mean, how the data ranges around the mean. And we can see if there's a significant difference, there should be no overlap between the data. But look, there's massive overlap because that goes down to 11.8, this goes down to 9, and they even overlap at the top end, the 13.4, the 17.8 goes above 16.6, so we've got overlap all over, top end and bottom end. So because we have overlap between the standard deviations, between the two means, it means there is no significant difference between the means. And the difference in those means, the fact that one is 16.6 and one is 13.4, is likely that is down to chance. The probability is that the difference in the means. Sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks. Question 6. So define the genome and proteome, worth 2 marks. Pause the video, have a go. Okay, the genome is basically all the genes or all the DNA in an organism or in a cell. So all the genes in a cell or all the alleles, all the DNA that is inside a cell, that is known as the genome. Now the proteome is only the DNA or the genes that specifically code for proteins, or you could say the full range of proteins coded for by the genome, or all the genes in the DNA that code for proteins. We know that not all genes code for proteins. For instance, introns are non-coding DNA. So that's not part of the proteome. The classification system used in the early 20th century grouped different species of bacteria according to the position and the shape of the flagella on bacterial cells, and by the number of flagella per cell. These were observed using an optical microscope. Each species of bacterium has a characteristic cell shape and an arrangement of flagella. These characteristics may be shared with other species with a genus. Within a genus, flagella are fragile, difficult to stain, and may extend from the cell at any angle. Consider the accuracy and limitations of the early classification of bacteria using the arrangement of flagella. So we're really looking at why is it not good to classify bacteria by the arrangement of their flagella. Well, we're just picking out the bits from the passage that are the limitations to this classification system. So they used optical microscopes, and we know that they have a low resolution based on the fact that the wavelength of light is only 200 nanometers, and anything smaller than that, it can't distinguish as separate objects. So the resolution of light microscopes is a limitation for classifying these bacteria. The flagella, we're told, are fragile. They can be damaged or broken. So what we think is flagella may not actually be flagella. Of course, not all bacteria have flagella, so it's going to be very difficult to classify some bacteria when they don't even have flagella, if the presence of flagella is your main feature for classifying the bacteria. So these are just some of the limitations to do with using flagella as a classification system for classifying bacteria. Okay, moving on to 6.3. We're going to look at suggest why several bacterial species have been renamed in recent years. So why have we in recent years renamed and reclassified certain species of bacteria. Well, we now have the invention of the electron microscope, both the transmission electron microscope and scanning electron microscope. They have better resolution and have enabled us to see things with modern staining techniques that have helped us reclassify some of the bacteria. Of course, there's also DNA sequencing we've had in the last 50 years. So the ability to sequence DNA, look at amino acid sequences, i.e. the primary structure of proteins, and do comparisons. And that has helped us group different species together and reclassify those bacterial species. Okay, let's zing on to 6.4. Welcome to Let's Get to the Marks. Just going to go through a quick A-level biology magnification question. So 6.4, figure 6 shows an image from an optical microscope of a single bacteria cell. There's figure 6. The bacteria cell is 2.3 micrometers long, excluding the flagellum. We don't need the tail. Calculate the magnification of this image and show your working. So for magnification questions, you need to know off by heart the equation I am. And you do a magic triangle with I at the top, A and M at the bottom. And that's where the A stands for actual size, or often called the real size. You can't see that with the naked eye. I is the image size, and that's what you actually see with your naked eye, like when you look through the microscope, or which is given to you in the exam to measure. M is the magnification. So let's rearrange this equation to find M, because that's what they're asking us to find. So we want to find M, we've got to do I divided by A. So we do I over A equals M. Now we go and substitute in the data that we already have. We have 2.3 micrometers long. Your eye cannot see something 2.3 micrometers long. That's very small. And anything that they're showing you is the image size. So we've got 2.3 at the bottom. That's the actual size or real size. And that equals M if we divide it by I. So where do we get our image? We have to actually measure across the middle of the bacteria lengthways. So from tip to tip, longest part. And if I measure this, this turns out to be 45 millimeters. So now the issue is that the micrometers and the millimeters are not the same unit. So I have a choice. I can either turn the micrometers to millimeters, or I can turn the millimeters to micrometers. Okay, so we can either do micrometers to millimeters, or what I prefer to do, let's rub that out, is turn the millimeters to micrometers, because you just times by 1,000. So times your millimeters by 1,000. So we end up with 45,000 micrometers on the top. And at the bottom, we've still got our 2.3 micrometers, as we didn't need to convert those units. And if I carry out that calculation, whack that in the old scientific calculator, you should get an answer around about 19,565, and that is the magnification. So that's how many times the image has been magnified compared to the real image, and that'll get you the 2 marks. Okay, let's get into question 7. So
yeah, baby.
That's a serious black screen. 7.1, carbon monoxide is released during incomplete combustion of fossil fuels. Figure 7 shows the dissociation curve for oxyhemoglobin when not exposed to carbon monoxide and when exposed to carbon monoxide, such that 50% of the oxygen binding sites are occupied by carbon monoxide, and we call that 50% COHB. We're shown on the graph the percentage saturation of oxygen against the PO2. Now, using figure 7, what can you conclude about how the exposure of carbon monoxide affects the loading and unloading of oxygen by hemoglobin? So first of all, I'm going to discuss the line which says 50% COHB, and what that's telling us on that line with the 50%, saying that when the carbon monoxide joins or binds to the hemoglobin, is associated with the hemoglobin, you do not get an oxygen saturation on the y-axis above 50%. So the maximum amount of oxygen the hemoglobin can carry once carbon monoxide is bound to it is 50%. So immediately that's going to score me a mark from pointing out that once carbon monoxide is bound, you can't get more than 50%. Whereas compare it to when no carbon monoxide is bound, you can get a much higher percentage of oxygen saturation. But also, this shows that when there is no carbon monoxide, there is a greater affinity for oxygen for the hemoglobin at high partial pressures. So on the x-axis where there's low PO2, that's like respiring tissues like muscles. What we find here is that the carboxyhemoglobin actually has a greater affinity for oxygen and is less likely to unload oxygen at respiring tissue. And that's a problem as well because our tissues need that oxygen for respiration, but there's a greater affinity of the carboxyhemoglobin for the oxygen. And so it's less likely to unload compared to normal hemoglobin that has no carbon monoxide, which has a lower affinity for oxygen at lower partial pressures, which is very useful because you want the hemoglobin to be unloading oxygen at those lower partial pressures like respiring tissue. So you want its affinity to change, whereas it seems like the carboxy hemoglobin is not unloading very well at low partial pressures, which means it has a higher affinity for oxygen than the no CO hemoglobin. And there we go. That should get you 3 marks, those comments. Not an easy question though, but you wouldn't expect that. You're at the end of the A-level biology paper. Towards the end, you get the difficult ones. Right, 7.2. The World Health Organization suggests that to avoid long-term health effects, COHB concentration should be kept below 2.5%. The WHO recommends that people should not be exposed to air with more than 10 milligram per meter cubed of carbon monoxide for more than 8 hours, or air with more than 30 milligram meter cube carbon monoxide for more than 1 hour. Scientists have been using a mathematical model to calculate the exposure of carbon monoxide that would result in 2.5% COHB in both adults and children. So the scientists suggest the WHO recommendations for carbon monoxide concentrations resulting in 2.5% COHB should be reduced. Evaluate. Okay, I'm just going to get straight into this one. Whenever you're asked to evaluate and there's all this scientific data, immediately check, have they controlled the variables? And when we look at the adults and children, we're not told what gender they are, what age they are, what ethnicity they are, and all of those we're not given any instructions on. So I can just pick up my 3 marks from ripping apart this study and saying, well, look, this could be wrong with it. Is exercise a factor? What about the exercise routine of the individuals? Is the mathematical model inaccurate? Also, looking at the data, there's been no statistical test. So if you're asked to evaluate someone's methodology, um, or the conclusion the scientists have made, you can make some 4 points, but a lot of the time you're going to pick up most of the marks from ripping it apart. So, yeah. Okay, moving on to 8.2, MIT-MAB or MITMAB acts as a non-competitive inhibitor of an enzyme called dynamin. Suggest how MITMAB can cause dynamin or dynamine to become inactive. So here's my enzyme with the active site and a substrate. Normally fits like a lock and key or induced fit model into that to form an enzyme substrate complex. Well, if it's a non-competitive inhibitor, it doesn't compete for the active site. Non-competitive inhibitors like MITMAB attach to a different area on the enzyme. Hopefully you know the name of this, but it's not in the AQA spec, but it's called an allosteric site. So you can either say MITMAB binds to an allosteric site, which is handy if you know that. Otherwise, you've got to say MITMAB binds to a place on the enzyme that is not the active site or other than the active site. So it just means any part of the enzyme that's not the active site. You don't need to know that key word in AQA, but they will give you the mark in the mark scheme for calling it the allosteric site. It saves you having to say that the inhibitor binds to a different part of the enzyme other than the active site. So you can slightly shorten down what you've got to write. And what does that do? Well, that alters the 3D structure of the active site. Well, alters the tertiary structure, the 3D structure of the enzyme, and therefore it alters the shape of the active site, and the active site is no longer complementary to the substrate. Now we need to use words like complementary because that's an A-level word, and they might not give you the mark if you just say active site changes shape, substrate doesn't fit. We want to use words like substrate is no longer complementary to the active site, or active site no longer complementary to substrate, and so less or no enzyme substrates can form ES complexes. Enzyme substrate complexes. So that's another key A-level word. So try to use A-level terms that you learn in A-level. Think, did I learn this in GCSE? Sometimes you get marks for GCSE terminology, but most of the time, if there is an A-level substitute for that GCSE terminology, you need to use the A-level version. Like words like complementary or enzyme substrate complexes. Coolio, let's zing on to 8.3. When active, dynamine has two functions. It stimulates cytokinesis. It inhibits cell death. So that enzyme causes cytokinesis. It causes cells to divide. It stops cells from dying. So you can imagine this enzyme is going to create a lot of growth in the body. And therefore linked to cancer and things. So scientists treated actively growing cultures of cancer cells with MITMAB. They incubated one sample of 2,500 cells, that's a lot, without MITMAB as a control, and 8 samples which each also had 2,500 cells and a different concentration of MITMAB. So they're seeing how the different concentrations of MITMAB affect cell growth by inhibiting this dynamine enzyme. After 72 hours, the scientists measured the number of cells in each sample. Figure 9 shows the scientists' results. A negative value for the proportion of control growth means that fewer than 2,500 cells were counted after 72 hours. So if it falls to a negative value, it means they have less cells than what they started with. So there's negative growth, essentially. The death rate is way outweighing the birth rate of cells, so to speak. Use all of the information given to explain the results. 3 marks, figure 9. I'm going to write my answer up here and not bother writing it down below because I need the graph in order to talk about it. So first area that I'm going to talk about on the graph is here where it's straight. And what does this say? There's no extra cell death here. There's no cytokinesis. Cytokinesis is still being allowed to continue, and there's no extra cell death. And what we're seeing is that at this point, low concentrations of MITMAB, we can see that it's had no effect overall on the enzymes at the moment. Why do we know that? Because you see where it says 1.0 on the y-axis, the proportion of the control growth is the same. And what that means is that these cells that are being treated with MITMAB are actually growing at the same rate as the cells without MITMAB. So it means the allosteric sites aren't all getting blocked. It means lots of the enzymes still have their functioning active site. And that's what we can learn from that. So there's our first mark. Low concentrations of MITMAB have not inhibited enough dynamin enzyme, so there is no change in cytokinesis cell death. Now we can look at the higher concentrations up to like 1000. We can see that the MIPMAP is inhibiting enough of the enzymes so that as a proportion of the control, we have a decrease in cell growth compared to those cells without MIPMAP. So yeah, some of the enzymes have been inhibited. Of course, that's permanent inhibition when you're blocking allosteric sites and changing the three-dimensional shape of the active site. So this is reducing the amount of cytokinesis and allowing cell death to occur. And so we have a decrease in the proportion of cell growth when compared to the control. Okay, that's our second mark. And then I'd really look at a third mark. So going all the way to the other end of the graph, you can see we've got a straight line again. So you're really breaking the graph down into different shapes and sections and interpreting what that means. So I'll go right to the right-hand side where there's very high concentrations. So if we go up to sort of 3,000 to 10,000, from there, that's like 3,000 to 10,000 concentration. So at the very highest concentrations of MIPMAP, we can see that there is no cell growth. How do we know there's no cell growth? Because we've got a straight line. So it means they increase the concentration from 3,000 till about 8,000, looking at that. And from between 3,000 and 8,000, they've doubled the concentration, obviously. 3,000 to 6,000 is doubling. And that has not changed cell growth. So this means that all of the active sites have been denatured. They've all lost their shape due to the allosteric sites being filled with the MIPMAP inhibitor. So I would say all the enzymes are inhibited at this point. So we have no increase in cell growth. It doesn't matter if we put 1 million concentration of MIPMAP micrograms per decimeter. It doesn't matter. We would not improve the proportion of cell growth. Okay, so that was the third mark. Now for the final part of this, we've got a little maths one. Whoopee! 0.01 decimeter cubed of MIPMAP solution was added to the treated cells. Now we need to remember that because I'm going to go back to the graph to calculate this. Calculate the increase in mass of MIPMAP in micrograms added to the cells to reduce the cell growth from equal to the control to 0.0 of the control. So we want to start at a point where the cell growth is equal to the control. And we want to finish to find our difference at where there is 0 cell growth when compared to the control. So what do we mean by equal to the control? Well, if you look on the y-axis, this says proportion of control growth. So at which point on that line is cell growth the same as the control? And that's going to be where it's 1, because 1.0 means that the cell growth with MIPMAP is 1 times the cell growth with the control, and 1 times 1 is 1. So that is when it is equal to the control. So times 1 as a proportion is equal to the control. When it is at 0 to the control, where do we find that? Well, we're going to find that with that dotted line. So what I want to do is work out the concentration of MIPMAP, so they're getting us to read the graph, and each one of these squares here on the graph, they're not equally spaced, and you probably find these annoying in biology. They're not equally spaced, but each one is worth 10 in that first section. So that is 70 micrograms per decimeter. Now I've started there, but you could have started at either of these two arrows. You could have started at the first arrow, and that is 30 micrograms per decimeter. It doesn't matter. They'll give you a range of answers in the mark scheme that will be correct. So my starting concentration is 70 micrograms per decimeter of MIPMAP, and my final concentration, it told me to work out the change in mass between equal to and at 0. So this is the 0, and the 0, you might be looking at that and going, oh, it's 1 square on, it's 1,010. It's not. We're going up in thousands in this last section. So it's gone from 1,000 to 2,000. Each section is worth a different amount. So you just have to check that. And so that's gone up to 2,000 micrograms per decimeter. So now we need to find the difference in the concentration. Well, actually, we've been asked to find the difference in the mass. So we can find the difference in the concentration, then convert that to mass, or we can convert these both to masses first and then find the difference. So in order to find the mass, when you've been given the concentration, you can use this equation, mass over concentration times volume. So in order to find the mass, I need to do the concentration times the volume. Now you remember I said remember 0.01 from the previous page. That's because that was the volume of solution. So we now know the concentration and the volume. So you do C times V gives you M, which is the mass. 70 times 0.01 gives you 0.7 micrograms. And that's mass, so it's not per decimeter, which is the concentration. Now we need to do the same for 2000. So we know the concentration when it fell to 0, the concentration of MIPMAB was at 2000 micrograms per decimeter. Times that by the volume, the same volume, 0.01, and we get 20 micrograms. Now we need to find the difference in mass. 20 micrograms minus 0.7 equals 19.3 micrograms, and that will get you the third mark. You actually get the first mark for just working out the two concentrations. And remember, the final concentration is always the same because there's only one place where the line crosses 0. But we could have started anywhere between these two x's up here. So anything between 30 micrograms and 70 micrograms could have been our starting point, and that would change our mass. So we're given a range of masses in the mark scheme from 19.3 to 19.7. That would have been the correct final answer, as long as you've used the correct starting zone. Because it asked us to start somewhere where the proportion was 1, where it was equal to the control. And here we go. Okay, question 9 is about the ELISA test. Oh my days. Dengue fever is a human disease caused by the dengue virus. Scientists designed an ELISA test to detect antibodies to the dengue virus in a patient's blood sample. Figure 10 shows a diagram of this test and some information about how it works. We got diffusion from X of the anti-human antibody with the enzyme attached. Essentially, that's just a monoclonal antibody coming down from X, and it has an enzyme attached that will react with a substrate and create a color change if you have the disease. And from Y, we've got the patient's blood sample diffusing upwards. Figure 11 shows the negative and positive results that were produced 20 minutes after the use of the test shown in figure 10. So we can see the negative one, you get a control line, but if you're positive, you get a control and a test line. Suggest what is on the test line T that creates the positive result. So what do we think is stuck on test line T? We're going to need a color change, right? And if you need a color change with an enzyme, what must be present? We know that the enzyme linked antibody is moving down, the ELISA antibody is moving down from the circle at the top. So what must be present on the T when it gets there that it changes color? And it's going to have to be a substrate, because the enzyme is attached to this antibody. That's my little Pac-Man enzyme there, attached to the antibody, and it diffuses down towards that line T. And on the line T will be a substrate. So if it gets there and it binds with the antigen, the immobilized antigen or whatever's there, not the immobilized antigen, but if it binds with an antigen that's in the patient's blood and it reaches that line and binds with the patient's blood, the timing will have to be right, then it is going to change color, that line. And it's going to bind with the antigen, and there's a substrate attached which will lock into the active site of the enzyme and be catalyzed and change color. So we need a color change. We need a substrate to bind with the ELISA enzyme and cause the color change. Also, the antigen that is complementary to the antibody has to be present, and that will get you 2 fat marks. So let's put that down here. The antigen must be present on line T to bind with the monoclonal antibody to form an antigen-antibody complex, but also we must have a substrate present on line T, because it's the substrate that will lock into the active site of the ELISA enzyme and create the color change that allows us to know we have a positive result by lighting up line T, purple, pink, blue, whatever the color change is. All right, let's zing on to 9.2. A line at C shows the test has worked. So just one reason why a line at C shows the test has worked. We'll look back up. We've got the ELISA enzyme diffusing down, and we also have the patient's blood diffusing up. If we get a line at C, what does this tell us? This tells us the patient's blood has made it past the T test area. And that means that it didn't contain any antigens if it got to C, and it just proves that it's at least gone past the test area, because you might not get a line on T and you might think, great, I'm not positive for dengue fever, but it might just be that the blood sample hasn't got to T. So by having a line at C that lights up when the blood reaches it, we know that the test has worked and the person doesn't have dengue fever. Right, figure 12 shows a flow chart for the anti-human antibodies, with basically how to make monoclonal antibodies. You did this in GCSE if you did AQA, and you should know this off by heart. So it says, suggests why the cells, the fused cells, allow continuous lots of production of monoclonal antibodies. Well, do you remember about the hybridoma at GCSE? That's the fused cell here. You don't have to call it hybridoma. But do you remember what it's made from? It was made from a cancer cell which rapidly divide by uncontrolled mitosis, and it was fused with a B lymphocyte from a mouse. In this case, we've got a rabbit, so it's fused with a lymphocyte, which can produce the correct specific antibody that you want to make lots of. Okay, so here we want to make the antibody so it was complementary to the antigens on the dengue fever virus, but that's neither here nor there. Your 2 marks are for saying cancer cell, uncontrolled mitosis, fused with a B cell that can produce antibodies. Yeah. Ethics peoples, evaluate the ethics of this. So what can we say? You're injecting rabbits, you're harming rabbits. So something about it being not good because you're harming rabbits. And then that's our negative thing about the production process, is that you harm rabbits. What's the positive? You make lots of monoclonal antibodies, and they're used to save lives with diagnostic testing in medicine, to find out if people have HIV, to find out if people are pregnant, to find out if people have dengue fever, people have COVID. So actually, there are really good benefits for medicine and the medical industry for diagnostic testing. And monoclonal antibodies aren't just used in tests like this dengue fever test. They're also used in like targeting cancer cells in chemotherapy and stuff, to deliver the chemicals so they only attach to the mutated cancer cells by binding to the antigens, and they carry a drug and that kind of thing. So basically, we just want to say that they're really good to be used in medicine. They can have lots of benefits, but at the same time, there's an ethical question about cruelty to animals and the way rabbits are being harmed or stressed. Right, moving on. Early identification of dengue fever can be difficult, as many other diseases produce the same symptoms. Early identification is important because people suffering with dengue fever can become ill very quickly and they might need hospital treatment. Scientists compared the effectiveness of 3 diagnostic tests for dengue fever: lab-based testing, current testing used in the doctor's clinic, and a new doctor's clinic test. So basically, you can turn up to the doctors, they test you, there's a current test, now we've got a new test when you turn up to the doctors, and then there's a test they have to send off to the lab. Let's compare the effectiveness. So number of cases of people, when they were 14, the lab picked up 10 of them. The current test, blooming useless after one day, didn't pick up any, and the new test 6. So we can see the new test is actually better than the current test when the symptoms have been between 1 and 4 days at picking up a diagnosis. And the new test is actually better than the lab-based test after 5 days. So scientists recommended the new test is used for the identification of dengue fever in all countries around the world. That's always a no-no, isn't it? All countries. It's not going to be the best thing for everywhere. Discuss this recommendation, use all the information given. So I'm going to go back up and have a little look. So what I would say straight off, let's make some points. Cover each row one by one and you'll pick up all the marks. So cover the first row. Time of onset is 1 to 2 days. So early on, someone's feeling ill, they've got the symptoms. Which test is best? So we can say, well, the lab test is best, but the new test is way better than the current test, right? The new test is better than the current test for early detection after 1 to 2 days of symptoms, but it's not as good as the laboratory test. Bash, you're going to get a mark there. After 3 to 4 days, let's cover that row. What can we say about the new test? The new test, again, is way better. It's how many times better? 4 times better than the current test, and nearly as good as the lab-based test. So another mark coming in. And then we want to make a conclusion about the final row. In the final row, it's equal to the current test. So the current test is equally good with the new test after 5 days. but they're both way better than the laboratory-based test. Now we need to say, like, would we swap it out all around the world in every doctor's surgery? Would you swap for the new test? You've got to think about, if you're bringing in a new test, you're going to need to train staff. So maybe they don't have the money, maybe it's a bit more expensive to do that than carry on using the current test. However, the new test is probably way cheaper than sending the results. It tells us here the results have to be sent to the lab. So that's way cheaper than the NHS or the health service in that country sending their results to the laboratory. So we need to say, look, and some countries may not have the scientists and the laboratories, so it might not be suitable everywhere to have lab-based testing, and those will be perfect for the new test where you have doctors. But then again, some places might not have the training or the people to be able to use the new test, and or even have enough doctors at the clinics. So they might not even have enough doctors at the clinics. In that case, they need the lab-based testing to get accurate results because they don't have the staff to carry out the new test. So there's kind of like quite a few evaluation reasons there for why it's not good all around the world, and there's definitely a mark to be picked up there. And you should hunt out things like that in the stem of questions when they make sweeping statements like, this should replace all the tests around the world, or this medicine can cure all people, or this medicine can treat all forms of bacterial infection, when they've only tested it on one. So always look out for those. They often have those in biology papers, sweeping statements that you can counteract and get yourself a lovely mark. Right, let's move on. So we are on to 9.6. The dengue virus causes damage to capillaries so that blood proteins move out of the capillaries into the tissue fluid. Explain how this would affect the return of tissue fluid into the capillaries. Right, so I'm going to do a quick little crusty drawing. No, I'm not going to do it down here. Let me do it up there. So we have blood from the left side of the heart going down arteries, breaking down into smaller vessels called arterioles, which I'm showing here in red. And then it goes into the capillary and it moves towards the venial end where it will return to the veins. But in between, it's delivering oxygen, glucose, and all the goodies to the cells. And water moves out into the tissue fluid because proteins normally cannot fit through the gaps in the capillary walls. They can't fit through those fenestrations. However, damage to the capillaries has made the gaps bigger. Look, I'm dragging some of the proteins down into the tissue fluid. Oh, I can't get that one. That's an annoying protein. Right, let's drag that one down. Okay, so the damage to the capillary walls allows the protein into the tissue fluid. Why does this stop water uptake, or how would this affect the return of water uptake? So you've got to think about water potential here. What will proteins do to the water potential of the tissue fluid? They're going to lower the water potential of the tissue fluid, and because they've vacated, they've left the capillaries, the water potential in the blood is going to go up. So instead of water moving back into the capillary down a water potential gradient, it's going to go the opposite. And even more water, we already lost water at the arterial end, but it means even at the venial end, even more water is going to leave the blood and enter the tissue fluid. And less water is going to be able to be taken back into the capillary by osmosis because the blood will have a higher water potential than the tissue fluid, which now has a lower water potential thanks to the proteins moving into it. And that's really your explanation. And I would imagine then people with this dengue virus, I imagine they get swelling in their legs and lots of sort of swollen limbs because of the inability of water and fluids to be put back into the circulatory system. And there we go. That is the end of question 9. Let's move on to the next questione.
Keep moving on, keep moving on, keep moving on, keep moving on.
Describe how a quaternary, I always have an issue with that word, I say quaternary, as quaternary protein is formed from its monomers. Do not include the process of translation. So the monomers are amino acids. How are they joined together? They're joined together by condensation reactions, going to get us a mark, which form peptide bonds, going to get us a mark. So there's 2 lovely marks there. Monomers, named as amino acids, joined together by condensation reactions that form peptide bonds. Of course, that forms the specific sequence, i.e. I've got circle, triangle, square, circle, circle, triangle. The specific sequence of the amino acid forms the unique primary structure of a polypeptide. Okay, let's zing to the second point. So next you need to think about how is the secondary structure formed. So think about your secondary structure. What happens to the polypeptides? Once they form that nice long chain, what do? Well, they fold. We get the first level of folding, and this is caused by hydrogen bonding between R groups and between the kind of like the ends of each peptide, which you can get hydrogen bonding between the chains like that. And so you can either have an alpha coil or beta pleated sheet, but you're going to get the mark really for secondary structure. You have hydrogen bonding. They just want some key points here, right? So we've already picked up potentially 3 marks. Super duper. Now, how does the tertiary structure form? Well, we get interactions between the different R groups. So my triangle will have a different R group to my square or my circle amino acid. And those different R groups, depending on the R groups, you might get ionic bonding or covalent bonding like disulfide bridges, or you might get hydrophilic and hydrophobic interactions. And this further folds the protein or polypeptide, giving it a tertiary structure. Of course, if it isn't a quaternary protein, then the final 3D shape is determined by the tertiary structure, because you just got one polypeptide and all of that bonding creates the final 3D shape. Of course, this question is about a quaternary protein, which means it has multiple polypeptides and they interact to form the final 3D shape that we call the quaternary structure. So a quaternary structure has multiple polypeptides. I'll show that with different colors in my cool little diagram here. Okay, got like a black polypeptide, a pink one, and then a kind of a green one, and they're all intertwined. And then we call that protein a quaternary protein. Okay, 10.2, describe the structure of DNA and the structure of a chromosome. So you would think at first you're going to get 3 marks for DNA, 3 marks for chromosome, but it's not weighted in that way. That's how you might assume, and you might actually only get 4 out of 6 here, and you couldn't be blamed for trying to do it like that. But there is more to talk about DNA than chromosomes, so hopefully you'll have waffled on more about DNA. So for example, DNA is a polymer made of monomers called nucleotides, so that's going to get us a mark. Then describing the nucleotide structure. So nucleotides, what are they made of? What are the 3 things? We've got a phosphate joined to a deoxyribose sugar, joined to a nitrogenous base. And then we could describe the bases to get some more marks. The nucleotides are held together, one nucleotide to each other in one single strand, are held together by phosphodiester bonds. Now, those phosphodiester bonds, going to get a mark for that funky word. Where are they located? They're located between the deoxyribose sugar and the phosphate group of the next adjacent nucleotide, not opposite nucleotide, adjacent. And then opposite each strand, DNA is double-stranded. So we're going to get a mark for DNA is double-stranded. We'll even get a mark for saying it's got a double helix, describing its shape. But we can then go into the complementary base pairing and explain that. So, i.e., each strand of DNA is held together by hydrogen bonds, complementary base pairing between adenine, thymine, cytosine, and guanine. Now, in the mark scheme, just a little word of warning, at A level, because most people have been introduced to CG, A, and T when they did triple biology at GCSE, they don't want to give you a mark if you say G to C, A to T at A level. That's how stingy they are, because they're like, hey man, people knew that at GCSE. So what you have to do here is you have to name it. So you'll have to go out the long way of writing guanine with cytosine, adenine with thymine, like that. And don't get your adenine mixed up and call it adenosine, because that's a common mistake, people to call adenine adenosine, because of course of ATP, adenosine triphosphate. Although the base in ATP is adenine also, it's just they call it adenosine. It changes its name when it's in that molecular formation. Right, so they want you to use the full names of ATGC, adenine, thymine, guanine, cytosine, and spell them correctly, even if I haven't. I'm not sure I have, I just haven't checked. Right, DNA is combined associated with proteins. What do you call the proteins that DNA are tightly wound, a bit like cotton around a cotton wheel? So they're called histones, and each strand of DNA has more than one histone that it's tightly wound around. And of course, later on in biology, you learn about how closely associated it is with histones, either increases the ability for transcription or decreases transcription. By allowing the machinery of transcription to get in and access the base sequences. So during mitosis, chromosomes, just a tiny little bit on chromosomes here. You only get like 2 marks for chromosomes, just mentioning that they're made up of chromatids and they have a centromere. I mean, yeah, bit of a bummer, that part to the question, but there we go. Mutation can result in an increase in genetic variation within a species. Describe and explain other processes that result in increases in genetic variation within a species. So, where do we get variation? You should know this off by heart. This should be the content that's on your revision cards. During meiosis, crossing over between homologous pairs. So crossing over, where does it happen between homologous pairs? That's where you get the chiasmata forming, the little bit where the chromatids wrap around each other and they snap off and swap alleles. So, for instance, my pink chromosome is going to swap with its homologous partner, the green one, and then we're going to get new combinations of alleles in the daughter cells, essentially. And so crossing over, that gives us some variation other than the random mutations that all things get, or all chromosomes or genes can undergo. Right, we've got independent segregation, which they kind of put on a same marking point as something called random assortment. Random assortment is it's randomized how they line up on the equator. And then independent segregation, when chromosomes pull apart, we don't know which one is going to which pole, essentially, like which bit is going to which pole of the cell, and so which bit is going to end up in which maternal or paternal alleles are going to end up in each daughter cell. We don't know which combinations, and we call that independent segregation. And then, of course, you've got random fertilization of gametes. You cannot just say random fertilization. You've got to say random fertilization of gametes. So we don't know which of these three sperms is going to get to the egg. That's random. Obviously, there's like 200 million sperm in a teaspoon. And yeah, we don't know which one will get to the egg. So they produce different combinations of alleles, and that's what gives us variation, is these different processes that all lead to different combinations of alleles. Okay, so one bit of advice. Some people, when they're doing biology paper 1 and only biology paper 1, like to tackle question 10 or the last question on the paper first. And the reason being because it's very content led. All of this stuff you can score high marks on. And should you run out of time, you would be absolutely gutted to have missed out on your marks for these processes that you've probably learned off by heart. And you'd much rather miss out on marks from things that you're probably going to drop marks anyway. So some people prefer to start this paper at the back and then go straight to question 1 after they've completed question 10. Just a thought for you. Don't forget, sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks.
You're still here? It's over.